Namaste everyone. Today in this lecture talk, I'll be discussing with you important questions for your internal test from paper 5, Genetic Engineering and Environment Biotechnology. Okay, so here I'm giving the collection of important questions. I request you to explore the answers for all those questions. Okay, and when you write the answer, important points are key words. Okay, are key points kindly underline, and after each answer, you must put double lines. Okay, now let us go through here. First main, answer the following. Okay, here in the first main, this carries 2 marks. First question, VAM, VAM. Okay, we need to expand what is this VAM. Then you need to write in 1 or 2 sentences the definition or what are VAMs you need to write. Second question, southern blotting. Okay, the southern blotting, even if this question can also be asked for 5 marks or 2 marks and even for 1 mark. Okay, you must go through the marks and then you should write answer. Okay, for 2 marks and 1 mark, you should, if you spend too much time, if you know the answer, if you spend too much time, and for 2 marks, if you write whole enter 1 page, then you'll be losing time. So for 2 marks, what kind of relevant points you need to write? No, only that points you need to mention. Okay, third question, GMO, you need to expand what is GMO and you need to define with example. Fourth question, give the differences between renewable and non-renewable sources of energy. Here you can give differences, like 2 differences you can give here. Fifth question, what is annealing? Okay. The next question, sixth question, lack Z gene. Seventh question, PAGE. You need to expand this PAGE and you need to write 2 sentences about PAGE. Eighth question, define restriction enzymes and DNA ligases with an example. Ninth question, northern blotting. Okay. 10th question, isoschizomers. Okay, you need to answer all these 10 questions, okay, for 2 marks. Now let us go through second main. Okay, answer any 2 of the following. Each question carries 5 marks. Okay, first one, give an account of symbiotic nitrogen fixation. Second question, explain the salient features of lambda phage as vector. Third question, write a note on southern blotting. Okay, here this question asked for 5 marks. So by keeping in mind this particular marks, you need to write the answer. Question number 4, write a note on PBR322. Fifth question, T4 DNA ligase. Here, you need to define what is this DNA ligase and what is the function of the DNA ligase. And here mention specifically T4. You need to mention this DNA ligase isolated from which particular organism. And you can also write here mechanism of DNA ligase, how the phosphodiester bond will be formed. Okay. Sixth question, explain bioleaching of gold. Seventh question, explain different vectors used for gene cloning in bacteria. Okay. Eighth question, write short note on agarose gel electrophoresis. Hope I'm making myself clear to you. Kindly make a note of all these questions and try to answer. Okay, and all these questions are very, very important for your practicals. Okay, during Viva also all these questions will be asked. So kindly pay attention. Ninth question, what are restriction enzymes? Explain their types. 10th question, explain the construction of recombinant DNA with an example. Okay. Third main question, answer any two of the following. Each question carries 10 marks each. First one, describe in detail the steps involved in PCR. Add a note on its applications. Okay. Second question, what is blotting? List the blotting techniques. Explain Western blotting. Third question, discuss the role of recombinant DNA technology in production of insulin. Fourth question, explain different vectors used in gene cloning in bacteria. Fifth question, mention different types of blotting techniques. Explain any two in detail. Okay. Sixth question, explain the construction of recombinant DNA with an example. Seventh question, briefly explain the recombinant vaccines, hepatitis B, and human growth hormone. Eighth question, write a short notes on PCR. Okay. Second part, DNA manipulative enzymes. Ninth question, briefly explain mechanism of DNA ligases and restriction enzymes. 10th question, give an account of the production of recombinant vaccines. 11th question, explain the steps involved in preparation of chimeric DNA. 12th question, explain bioleaching with respect to copper and uranium. Okay. So all these 12 questions you must explore. Then you will be able to write your internal test very successfully. Okay. Dear students, do you know the differences between success and failure? Try to answer. What do you mean by success? What do you mean by failure? Think. Okay. All right. Doing is success. Not doing is failure. It's very simple. I'll repeat for you. Kindly focus. Pay attention. Be a good listener. Listening skills is very important during your student life. Doing is success. Okay. Not doing is failure. So, be always a good performer. Okay. If you're doing something in your work or doing your study or during your student life, then that will be your success. If you're not doing, that's a failure. Okay. So it's very simple. Okay. Doing is success and not doing is failure. Even Baswana also explored that, right? Kayakave Kailasa. You must do something. Next, fourth main, answer the following. Each question carries 1 mark. First question, what are Taq DNA polymerase? Second question, expand EDTA. Third question, what is symbiosis? Fourth question, what is electrophoresis? Fifth question, what is ectomycorrhiza? Sixth question, name any two bacteria used in production of biofertilizers. Seventh question, what are cosmids? Eighth question, Western blotting. 10th question, what are primers? 11th question, what is annealing? This question asked for even for 2 marks, and here we have asked here for 1 mark. 12th question, who discovered enzymatic method of DNA sequencing? Hope I made myself clear to you. Go through all these questions and explore all these questions. Okay. I'm Dr. Pratibha KS, signing out for today. I'll be back with some more videos. Stay tuned with me and thank you for watching this video.